let's talk about something truly fascinating. We're going to dive into this incredible and honestly pretty surprising connection between a little piece of a yeast cell wall, yeah, like from baker's yeast, and our body's own blood production factory, the bone marrow. It's a bit of a scientific detective story, and it reveals an amazing partnership happening right at the cellular level.
And that really is the central mystery we're unpacking today. I mean, how can a simple sugar from yeast possibly help our bodies rebuild something as critical as our blood supply? It sounds a little wild, I know. But the science here points to this really elegant biological strategy. So how on earth does it work?
Okay, first, let's set the stage with our main player, the bone marrow. The best way to think about it is like this super busy, high-tech factory tucked away inside our bones. It's constantly manufacturing all the essential parts of our blood. And while it's incredibly powerful, it's also surprisingly vulnerable to damage from things like radiation.
And now for the hero of our story, and it is a very unlikely one, beta-glucan. We're talking about a type of sugar molecule that comes from the cell wall of common baker's yeast. So for our purposes, let's give it a name, the surprising ally. And you are absolutely about to find out why.
So to solve this whole mystery, the first thing scientists had to figure out was, how does the bone marrow even signal for help when it's in trouble? Turns out, our body has a kind of built-in emergency flare gun.
So, picture this. The bone marrow has just taken a hit, say from a dose of radiation. The whole area is damaged. The very first thing that happens is the local environment starts sending out these chemical distress signals. It's basically screaming, "Hey, we need a repair crew over here, stat!"
And what is that distress signal? Well, it's a little protein fragment called IC3B. When there's an injury, this molecule gets splattered all over the structure of the bone marrow. Let's just think of it as a homing beacon. It's not doing the repairs itself, it's just lighting up the exact spot where the damage is.
So what's lighting the beacon? Well, a 2006 animal study published in the journal Blood found something pretty amazing. It's our body's own antibodies that recognize the damaged tissue and kick off the process to plant these IC3B beacons. This is what flags the area for the arrival of the cavalry, our hemopoietic progenitor cells, the stem cells that build new blood. Okay, so the beacon is lit, but the next logical question is, how do the stem cells even see it, and what happens when they get there? This, my friends, is where our surprising ally finally makes its grand entrance. I mean, a homing beacon is great, but it's totally useless if the rescue team doesn't have the gear to see it, right? Well, these stem cells need a very specific receptor to be able to lock on to that IC3B signal. And, of course, they have one. It's called complement receptor 3, or CR3. And what's interesting is that only about a quarter of these critical stem cells have this receptor on their surface. And this receptor acts just like a docking station. It's perfectly designed to connect to that IC3B homing beacon.
And this is exactly how it all comes together. Step 1, a stem cell shows up at the injury site. Step 2, its CR3 docking station latches on to the IC3B beacon. It's locked in place. But here's the key, it's just waiting. Then comes step 3, soluble beta-glucan from yeast shows up and binds to a different spot on that very same receptor. This primes the cell. It's like flipping a switch to on. And the result? Step 4. The now primed cell gets the green light to start multiplying and kick off the whole repair process.
And here's the proof in the pudding. In that same 2006 animal study, researchers looked at mice that had been irradiated. As you can see from the chart, the group that received an oral form of yeast-derived beta-glucan had a much faster recovery of their white blood cell counts compared to the control group. They were literally rebuilding their blood supply faster, all thanks to this priming mechanism. But hold on, because that's not the whole story. Not even close. Just when scientists thought they had it all figured out, more research revealed a second, totally different way our surprising ally helps out. See, it doesn't just supercharge the workers already on site, it actually calls in a whole new army.
So yeah, priming the stem cells that are already there is a huge deal. But then a follow-up animal study in 2008 uncovered a wild new twist. It turns out this yeast-derived beta-glucan has another trick up its sleeve. It actively mobilizes new stem cells, getting them to leave the healthy parts of the bone marrow and jump into the bloodstream.
So how does it do that? It uses an enzyme called MMP9. And the absolute best way to think about MMP9 is as a pair of molecular scissors. See, stem cells are normally tethered down inside the bone marrow,
MMP9 comes along and just snip, snip, it cuts those tethers and sets the stem cells free to travel wherever they need to go.
And this 2008 animal study laid it all out. What they found was that treatment with a specific kind of yeast beta-glucan made the bone marrow cells release these MMP9 molecular scissors, and that, in turn, released a whole wave of fresh stem cells. And the proof? In mice that were genetically engineered so they couldn't make MMP9, the effect was completely gone. But here's the real kicker. This entire process was totally independent of that CR3 docking thing we talked about. It's a completely separate strategy.
So now we have these two completely different pieces of the puzzle. One is all about beefing up the repair crew that's already on site, and the other is about calling in brand new reinforcements. And when you put them together, you see the full, elegant picture of what's really going on.
And this is the big reveal, the aha moment. Yeast beta-glucan is playing two different games at once. On one hand, it's boosting the stem cells that have already docked at the damaged site. That's the CR3-dependent part. And on the other hand, it's triggering the release of a whole new wave of stem cells by activating MMP9. It's this beautiful, efficient two-pronged attack to speed up recovery.
So, the big question is, what does all this really mean? Well, this preclinical research paints a truly fascinating picture of how a natural compound could potentially support one of our body's most basic, most critical repair processes.
You know, you don't even have to take my word for it. The authors of that original 2006 study said it perfectly. They wrote that their findings suggest a novel role for beta-glucan and the restoration of hemopoiesis following injury. This wasn't just some small effect. It was a whole new way of thinking about how to support this process.
Okay, so let's just do a quick recap. What we've seen is that yeast-derived beta-glucan seems to help our blood factory recover with this clever dual strategy. It boosts the workers already on site, and it calls in an army of reinforcements. Now, and this is really important to remember, these incredible findings are based on preclinical work. That means studies done in the lab and on animals. This is the foundational science that really points the way for future research. And that really just leaves us with this one final amazing thought. Isn't it incredible to think that a key to helping our bodies recover from some of the most profound injuries might not come from some complex synthetic drug, but from a simple, natural piece of the wall of the very same yeast we use to bake bread? It's just a powerful reminder of the sophisticated solutions nature has been working on for millions of years.